Hey everyone, welcome to this second part of your 4.1 module. I had made a note before to film this portion instead of giving it live so that you can pause and write down the information that you need to. So this part of the presentation is going to be kind of similar to functional groups in the sense that you're going to need to know the names of these and what they do. And not all of them, but a good chunk of them. So taking your time to be able to record their names and what they do, we call that function in biology. And then also having some note of what their structure is. So how they're built, how they're put together. Because in biology, we say that form follows function. So form, what it looks like, follows what it does. So if you know what it does, how it looks like should allow it to do its job. But if you also look at something, you can also start to say, okay, it must do this because it's shaped that way. So this presentation will go through a breakdown of what we call cell organelles. So these are the bits of machinery in eukaryotic cells that carry out all the tasks. So, eukaryotic cell for you at this time, you should be thinking of us as animals. You should be thinking of plants. You could be thinking of protists, what you were seeing in the pond water. And then you can also think of fungi like yeast. So, all four of those kingdoms will have some combination of these organelles here. So, they delegate certain tasks to these different organelles to carry out the overall life function. So the way that I have broken this down is very similar to our textbook. So if you're going in and following with our textbook, I thought it might be easier if it was like, okay, they're kind of in a somewhat similar order. But at the same time, I know what we do need and don't need to know for the AP exam. And so I've shortened what you would do as you were if you were reading the book alone. So your goal is going to be to develop a working knowledge of the structure and function of each organelle. So these notes will help. You'll also be diving into one. You will become one for a dating game, speed dating, that we'll be doing in here, and I'll talk about that more in class. So what I have here is I've separated it down into essentially 6 different tasks. So these are going to be tasks that the cell needs to accomplish in order to survive. And so we have protein synthesis. So proteins do virtually everything for you, allow you to, if you think of anything that you do very well, it's because of proteins involved, likely hundreds of them. And so our nucleus, our ribosome, and our nucleolus, yeah, it sounds like nucleus, but it's a little bit different, are going to be involved directly in starting that process. So synthesis means to create. So protein synthesis. Endomembrane system, big word. So you probably understand the word membrane, and you should be thinking at this time, or I'll help you get to that point of thinking, lipid bilayers. So we had looked at that back in our biochem unit, that molecule that had the polar head and the hydrophobic tail. So these are going to be membrane. They're going to be surrounded by those phospholipids. And endo is a prefix that means within. So this is going to be a membrane system within the cell. We have the endoplasmic reticulum, and it's one organelle that has 2 different regions. It has a smooth and a rough region, and we'll talk in a minute about what makes them the difference. We have the Golgi apparatus, sometimes called the Golgi body. We have the lysosome. We have the vacuole, and we have the endosome. So we have a lot of different bodies that make up the endomembrane system. So they're going to be involved in transport, primarily transport and storage. Energy production. Our powerhouse of the cell is going to be there, the mitochondria, but also the chloroplast. And you would have seen these in our plant cells during our rotations. They were the little green beans that you saw within the cells. The external membrane system is going to be the plasma membrane, and in some of our kingdoms, the cell wall. Absorption, so taking things into the cell, and motility, movement, are going to be microvilli, flagella, and cilia. And then finally, our internal structure, so kind of the bones of the system, you have a cytoskeleton. And so, really, what you have with the cytoskeleton is going to be centromere and centrioles. So, they're going to be almost like steel girders that are across the middle of the cell. And you have a controller in the center that can reel it in to pull things closer to the nucleus, further from the nucleus. So, it's kind of like a construction site in a sense. And then finally, the goo that surrounds everything will be the cytoplasm. So we're going to jump in and just go one by one here. So according to the previous slide, so protein synthesis was the first one that I introduced on the previous slide. So here we're going to be looking at the ribosome, the nucleus, and the nucleolus. And so in each of these slides going through where we needed it, I also put an image of a close-up of what they look like. And then in each table here, I have the structure, so what it looks like, and the function, what it does. So we're going to start here with the ribosome. So the ribosome is where the finished protein product is going to be made. So this is where the primary structure of a protein occurs. When we're pulling, those 20 amino acids and starting to string them together using peptide bonds, that is going to be happening with these little blue pearls coming up right here. How does it know which one to make? How does it know which amino acid to plug in when it does? We'll go into greater detail with this probably around in February. But you have an RNA sequence that's being fed through the ribosome. And as it's being read going through the ribosome, this code in here will say, let's plug in a glycine right here. Let's plug in an aspartic acid. Let's plug in a cysteine. And so that is that is how the ribosome is going to work. So the ribosome is actually made of two subunits out of RNA. So it's an RNA-based organelle. So it has a large segment and a small segment down here, both of which are going to be made out of RNA. And they're bound to the endoplasmic reticulum. So we'll talk about the ER, the endoplasmic reticulum, in a moment. But you're going to find these right outside of the nucleus in the cell. And curiously enough, we'll also find them in the mitochondria. So that'll come up in our conversation a little bit further. So the mitochondria are going to have some ribosomes also. But the majority of them we're going to find right outside of the nucleus attached to the endoplasmic reticulum. And again, their whole job is going to be the assembly and folding of proteins. So I had said that we have a RNA code that's being or arrives at a ribosome that's going to allow the ribosome to know which of the 20 amino acids to put in place. Where does that come from? Well, that's going to come from the nucleus. So the nucleus is going to be an organelle that's surrounded by a membrane with pores. And what's going to go through those pores? Well, some of this RNA, because it has to get out of the nucleus and to the ribosome. And then you also have some proteins that are involved in the process going back and forth. And it's going to also be linked to the endoplasmic reticulum, okay, which should make sense. If the code for the proteins is here in the nucleus, we want where the ribosomes are attached to the endoplasmic reticulum to be right there. We don't want our RNA signal that's coming out of the nucleus to have to go a long distance in order to find one of these suckers. We want them right outside of the nucleus. So the nucleus is going to hold chromosomes, which are DNA. And so now you can kind of get a picture of the full process of how we make a protein. In the nucleus, we have DNA stored. When we need to make a protein, we have a middle step called mRNA. It's an RNA strand. The M stands for messenger. It's going to be the message that's sent from the nucleus to the ribosome. Then this mRNA will go to the ribosome in order to make the protein. So the nucleus and the ribosome are going to be really closely related in terms of function, because one is going to store the code for what proteins need to be made, and one's going to actually get her done and actually crank out those proteins as we go. The nucleolus is going to be this really dark ball in this diagram at the center of the nucleus here. And this is where ribosomal RNA is stored. And this will be used to make the ribosomes. So it is its own little kind of factory. So when it's time to replace our ribosomes that are doing all the protein folding, they're busy little critters. Um, the code for doing that is stored right in the center of the nucleus. Okay. So for protein synthesis, we have the ribosome that's actually going to fold or is going to actually start those structures, primary, secondary, tertiary. You learned about those. The code for making the protein will be held in the control center, which is the nucleus. And the nucleolus is going to have all of the ingredients for making more ribosomes when that is necessary. The endomembrane system. So this will be a couple of different slides for us. You can see membrane. We see a lot of folds in structures like this. Okay, so we'll start with the endoplasmic reticulum, and then we'll work our way to the Golgi. The endoplasmic reticulum is going to be a large network of membrane-bound tubes and sacs connected to the nuclear envelope. So our nucleus is right here. Remember, we're storing our DNA here. And our endoplasmic reticulum is going to be linked to the outside of that nucleus. And if you see all these little bumps here on the ER, those are the ribosomes. So our ribosomes are going to be attached to the ER right outside of the nucleus. So we don't have to send that RNA signal very far in order to start making those proteins that we need. So there is a section, we kind of separate the ER. The ER is super important, and it kind of has a jack of all trades. It seems like every day we're finding something else that it does. So we separate into two regions, and it's based off of how it looks under really, really strong microscopes. We have the rough ER in this section right here that I'm circling, and we have the smooth ER up here. And the difference is whether or not the ribosomes are there. If the ribosomes are there, it almost looks like Braille under a microscope. And so we've just called it the rough endoplasmic reticulum. So that's where the ribosomes are. And then we have a smooth region that is a little bit further away from the nucleus. You're like, further away? The nucleus is right here. But if you look, it's almost like a maze or a tunnel. Here is where you enter out of the nucleus. But then you go through all of this and around until you get to the smooth endoplasmic reticulum at the end. So the smooth will be further from the nucleus, which makes sense because the rough section with the ribosome should be right next to the nucleus, because then that RNA, which isn't the most stable molecule in the world, doesn't have to go too far. So to summarize what they do, the rough ER, it's going to aid the ribosomes in protein synthesis. It's going to be the anchor. It's going to hold them in place. Then once those proteins are made, the endoplasmic reticulum can start to modify or add on to those proteins. So we can tack on a carbohydrate group, and if we do that, we would call it a glycoprotein. Glyco can mean sweet, and protein meaning protein. So we can create glycoproteins at the site of the ER. In the smooth region, okay, so once we have made our protein and we've started to modify, we need to move them out of here. So it's going to be a part of uh, kind of the moving delivery system of these proteins. Is they're going to, um, once they have been made at the ribosome and perhaps modified, not all will, some will, in the endoplasmic reticulum, then they need to move through this maze and will find their way to the smooth ER, where they then will be shipped out of the ER onto their next path. But the smooth ER, aside from proteins, if we look at some of the other macromolecules, we know that lipids are synthesized or made there. We know that carbohydrates are metabolized there. Okay, so kind of the making and breaking of those bonds within carbohydrates. So think glycosidic bonds if you're going to link it back to the last unit. Calcium can be stored there. They also can detoxify, meaning take things out of the cytoplasm, the goo around it, in order to maintain pH or to remove things that might otherwise be unhealthy for the cell. The Golgi apparatus is this weird kind of membranous sac structure right here. So its main deal here is going to be receiving the proteins after they've left the smooth endoplasmic reticulum. So after they leave the smooth endoplasmic reticulum, they're going to be really put in little vesicles that we call endosomes, and those little vesicles will, kind of like escape pods in a sci-fi film, will bring them to the Golgi apparatus. Once they're there, they can be modified for the last time. And then they're shipped off to where they need to go. So at the Golgi, this is kind of like a FedEx center in a way. It's kind of like it receives all of these packages called proteins, and this one needs to leave the cell, and this one needs to go back to the nucleus, and this one needs to go wherever. The Golgi is going to help direct that traffic so that the proteins know or are sent to the right place. We're still trying to figure out a little bit more about how things arrive and depart from the Golgi. The Golgi is actually the most recently discovered organelle. I think it was the 1970s. And for you, that seems like ancient history, but that was right before I was born. And we know that there's something with polarity that brings in and releases these vesicles from the Golgi. You do not need to get into the weeds, but just showing you, we don't know everything about the cell at this time. So there's still lots of room for discovery as we go through. All right. So some other organelles in the system that are considered a part of this endomembrane system would be the lysosome, the vacuole, and I just mentioned the endosome. We'll start there. So the endosome is going to just be a vesicle that's surrounded by a phospholipid bilayer, and it's going to carry things around. Okay, so it's going to carry things between different organelles. It's going to carry a lot of stuff if it's coming into the cell to the lysosome so that it can be broken down into smaller bits first before your cell decides what to do with it. And in this slide right here, so this would be an example of an endosome. This would be an example of an endosome. So really, it's going to be a phospholipid bilayer protecting or surrounding something that it's carrying within it. The lysosome is going to be surrounded by a phospholipid bilayer, and it's going to contain extremely strong acid. So if you need to digest, that word digest goes with lysosome. The lysosome is going to be involved, and they're only found in animal cells. So this is going to be a way to break down some of your food that you're eating into the monomers that you need for rebuilding them to do all of the magical things that your cells do. So the lysosome is kind of like almost a little stomach for our cells. It's going to break things down into smaller pieces. So your cells can choose how you're going to rebuild that back up. It also has a really interesting function. If there is a white blood cell, you saw those in your slides. They were in the blood cells. They were the one that had like the purple nucleus in them. Those white blood cells will circulate around. And if they detect something's off with a cell that it's kind of in contact with, that white blood cell might say, this ain't right. The cell seems off, could be cancer, could be infected with a virus. Um, we're, I'm going to signal you to die so that you are not going to be a problem to spread any problems. And so, if that signal is sent by a white blood cell, all of the lysosomes in the cell will rupture and open, and a thing called apoptosis will happen, cell death. So there was a time when this was first coming out, when this was discovered, that the lysosome was sometimes referred to as the suicide sac, because it is how the cell will do apoptosis, really to kill itself in order to protect all of the other cells around it. So, anytime that you hear anything about digestion, a cell not being able to digest something, whatever, it's going to be tied to the lysosome. And the lysosome tends to be something they really are interested in on the AP exams. So, this is one that you're going to want to know. A vacuole. So both animal and plant cells are going to have vacuoles, but they're going to look a little different. So they're little vesicles surrounded by phospholipid bilayer, and they will store things. It's kind of like little closets in the cell. They can store water, nutrients, or waste products until a lysosome is able to break them down a little bit more. And so in animals, we have really small vacuoles, mainly for storing small amounts of water, nutrient, and waste. But plants, on the other hand, will have a huge central vacuole within, if you look right here, within the middle of the plant cell. In fact, it's the majority of the cell. Now, water is necessary for photosynthesis. And so every cell that's going to participate or do photosynthesis needs to have water readily available. And so you want it right there in the cell. So the central vacuole is going to be kind of like a water tower, water reservoir for the plant cell. In fact, when this vacuole drains because there isn't enough water being fed into it, so you take water in through the roots, goes up to the cells, and will swell the cell by putting it into the vacuole. If this depletes or drains, the cells shrink, or it's called plasmosis. And that's why plants wilt. So if you don't water plants, they wilt. Well, why do they wilt? Well, their central vacuoles are drained of all of their moisture. And so the cells shrink, and all of them shrink together. You're going to see a wilting phenomenon. So the endosome, they're all going to be little blobs surrounded by phospholipids. The endosome is going to be transport, moving things from here to there to where they need to go. The lysosome is going to be for digestion. So it has digestive juices, really, in order to break down things that are complex into simpler forms, or to get rid of things that the cell doesn't need anymore, or to actually open up and destroy the cell itself. And the vacuole is going to be like a closet. So even though they're all surrounded by phospholipid bilayers, their functions are going to be different. Energy production. So you've definitely heard of the mitochondria before. And it has some really interesting characteristics, as does the chloroplast. In fact, evidence shows they come from a different place. Mitochondria, they're going to be double membrane bound, meaning that they're going to have two membranes that surround them. The inner membrane is going to have folds, and there's going to be a fluid in the middle called the matrix. So if we look at this structure right here, kind of this brown outside is going to be the outer membrane. The inner membrane is going to be this kind of folded area. And then the matrix is going to be in the middle. Okay. But you're also seeing ribosomes and DNA. Yeah, I just told you the DNA is in the nucleus, so that's a little bit curious. We'll come back to that in a moment. What does the organelle do? ATP. We're moving to a point in a year where you're going to be hearing this acronym in your sleep. So ATP is adenosine triphosphate, and so this is going to be the energy molecule. So if you want to make energy, you're making ATP. And the process that does it is cellular respiration. Respiration, you might think, oh, it's like, isn't that when you breathe? Yeah. Okay. You breathe for this process. So the air that you're breathing in is used for generating ATP at the mitochondria. And when you exhale, that's the waste product from your mitochondria that have done their job. And that's going to be more in our next unit. Chloroplasts have a similar structure to the mitochondria. They have that double membrane. Inside, they have a fluid. I'm going to move me up here. They have a fluid that is referred to as the stroma. They have thylakoids, so they almost look like little. Well, I don't know if you've ever had the candy bottle caps before. It's an old time candy, but kind of like little pancakes in a sense. And a stack of them together is called a granum. And these are going to be like solar panels. So the whole point of chloroplasts is to make glucose through photosynthesis. So it's going to use these solar panel type structures to capture sunlight in order for that to happen. And the water for photosynthesis is going to come from that central vacuole. And you even saw stomata underneath the microscope, and that's where the carbon dioxide is going to come from. And there you have all the ingredients for photosynthesis right there. So things are starting to come together a little bit, and that always is a nice thing. So photosynthesis and cellular respiration are going to be what we call reverse reactions. So one is going to produce glucose and one is going to break it down so that we can get our energy. And this diagram essentially is all of Unit 5. So all of Unit 5 will be about energy production. But yeah, both mitochondria and chloroplasts contain DNA and ribosomes. So when scientists discovered that, we had a little bit of detective work to do. Like, why would that be? None of the other organelles, aside from the nucleus, have this type of thing going on. And so where we arrived is this really cool theory. And again, theory means we have all the evidence supports it, none disprove it. And this is an important theory. This is something that will be on the AP exam and your class exams too. So both mitochondria and chloroplasts, sometimes called plastids, were once unicellular prokaryotic cells of their own. So think of some of those protists that you saw swimming around in the pond water. But at some point around 1 million years ago, an animal-like protist engulfed a prokaryotic cell. Think of the bacteria that you saw, resulting in the mitochondria. So you had one cell eating another cell, and the prokaryotic cell started to provide its own function, which is energy. And so what you have now is you've harnessed a source just for energy. And without energy, there would be no advancement or complex life that would develop. There just would not be enough energy for that to happen. So once we have the mitochondria forming, then one of those cells likely engulfed a photosynthetic, so something that can do photosynthesis, thing called cyanobacteria. So it is a critter that can do photosynthesis. And the result was the chloroplast. So plant cells, this would be their ancestor right here. Okay. This would be their ancestor. And our ancestor would be this one right here. So plants are going to have both chloroplasts and mitochondria, whereas we are going to have just mitochondria.